This presentation will be about the basics, biomechanics and biology. And when we talk about biology, a lot of times we would use what is called a structural organization approach. So we go from the large level to the smaller level. In this case, it would be from the body and the organism to a tissue level, and then further down to the cell level, and then to the molecular level, which I'm not going to talk so much about. And I will use this exact same approach also now for my presentation. So for the spine, what we would like to start with is the systemic level. So this is basically the largest level lower than the body. So in this case, it would be the entire spine. And when we talk about the spine, what is important to understand is, and that is probably clear to all of you, that it's the backbone of the musculoskeletal system. It is, on the one hand, responsible for load carriage. So it carries the weight that is above it, which basically means the head, but also the pelvis and so on, and it transmits these loads then to the lower parts of the body. And maybe even more importantly, it is also very crucial in trunk mobility, and that is related to the structure of the vertebra and the intervertebral discs. When we talk about the spine, what comes to mind obviously directly is disease-related questions, and the disease that is most relevant for the spine is low back pain. It is very relevant because it has a very high lifetime prevalence of around 50 to 80%, and a high recurrence rate. It is, and that is a big problem for the health society and the healthcare systems, it is something that affects mostly the working population, so it is therefore why it has such high indirect costs associated, but of course also very high direct costs. It is therefore one of the most costly diseases of all. And when we talk about diseases, what is always very important to understand is the biology and the biomechanics, because that is the basis for the development of novel and more targeted treatment options. Unfortunately for back pain, the biomechanics and the biology are not so well understood. There has been a lot of research going on over the past years and decades. Biomechanics is probably more understood, biology not so much, which is also the reason why we don't really have any perfect treatment for these patients with back pain. They're usually either not very effective or quite invasive with a lot of side effects and complications, and the costs are very high. Now, the fact that the spine is the backbone of the musculoskeletal system becomes very, very evident when you think about the loads that occur in the spine. The loads are up to 5,000 Newton, which translated to kilogram would be 500 kilograms, and that is during normal daily activities. It's not something that is uncommon. It's during your normal lifting activities and so on. And that shows how much load the spine actually has to carry. And what we're going to do is we will look at the different structures of the spine, vertebra disc, ligaments, and see how these contribute on the one hand to carrying these loads, but also to stabilizing the spine. There have been quite a few studies that looked at loads in the spine. In the 1960s, that was done by Nachemson et al., later by Wilk et al., and then more recently by Sato. And basically what these studies all did is they put a pressure sensor into the intervertebral disc in a needle form, and then the subjects, the volunteers, had to perform different tasks. These could be posture tasks, so just bending backwards a little bit, but still sitting, but it also could be really activity tasks like lifting a heavy weight. And on the right side, you can actually see the different tasks, all calculated relative to the sitting position. And the sitting position is then put to 100%, and you can see that when you bend over and lift a heavy weight, like a beer case, then the loads on the spine and the disc, they really increase a lot, up to 5-fold more. And this in the end results in the fact that the intradiscal pressures can be as high as 3 megapascal, which is very high if you think about it. So the structure of the discs need to compensate for that and need to be able to withstand these forces. And if they don't, then that's exactly what you would see in the clinic a lot of times, where you had patients with a disc herniation, where you have tissue failure, and that is often associated in the end with mechanical loads being applied to the intervertebral disc and the spine, but it's not necessarily the underlying reason, it's just the final contributing factor. Now, what has been done more recently is actually very interesting. There are studies by the group in Berlin, the Charité, where they use implants that can actually detect the loads. So it's basically load sensors that are implanted instead of normal implants. And what they have been doing is create a database that is open source. You can get access to all that information. But they measure the three-dimensional loading of the spine in all kinds of different activities. And because these are implants, of course, this also includes activities such as skiing or other sports activities. So if your patients would ask you, what can I actually do? What is good for the spine and the disc? Where are the loads very high? That would be a very good source to look at to get some information. Now, if we move one level down, we would arrive at the organ level. And for the spine, that would be the functional spine unit. This is kind of related to the term that is called motion segment. That's probably something that you've heard before. So it's basically a vertebra, a disc and a vertebra, but then of course also the ligaments and the muscles and the facet joints. And it's the smallest unit of the spine that kind of resembles the entity of the spine biomechanically, but also biologically. Now, if we look at the different parts of the spine, we can start with the vertebra because they are biomechanically very important. And here on the left side, you see the vertebra in the cervical, the thoracic, and the lumbar area. And the first thing that you will notice is the size difference. They do become bigger once you get lower in the spine. And that makes sense because it correlates also to the increasing loads that you have in the lower parts of the spine, in the lumbar area. And these loads are on the one hand related to the fact that this part of the spine has to carry more weight because the lower you get, the more upper part of the body you have. So in the cervical area, these vertebrae would only need to carry the head, but then of course the lower you come, the more it is. But it also correlates with the fact that in these lower areas you have more muscle forces. And these also put load on the spine and the vertebra, so they need to be bigger and more stable to be able to take over these loads. What you can also see is here on the right side, the two different structures of the vertebra. So on the one hand, you have the posterior, and on the other hand, the anterior area. Here the posterior area is carrying most of the load, and that is why it has a very optimized shape. It's elliptical, and that makes the load able to distribute over the entire area so that there are not so many load peaks occurring that can then be a cause of fracture. And on the other hand, you have the processes here, and they stick out quite a lot. They don't carry so much load, but they have the muscle attachment and the ligament attachments, so they are very important for stabilizing, but also for mobilizing the spine. They do stick out so much because that creates a larger arm, like mechanically speaking, so the ligaments and the muscles can actually act more efficiently in that way. Now, if we go more inside of the vertebra a little bit, I would just like to point out the structure of the vertebra. It's something that you've probably learned already at some point of time at university. There is two different areas of the bone, which is the cortical and the trabecular bone. The cortical bone on the outside, it's the compact bone, and the trabecular bone in the middle. And the trabecular bone is also the one that is most affected by osteoporosis. So during aging, or also with hormonal differences in females, you will see a loss of trabecular bone predominantly, whereas the cortical bone stays more or less the same. And this is actually a picture of here a more healthy person, a younger person, and then of an aged person, where you see this decrease in trabecular and a thinning, which then leads to higher pores or larger pores and less mechanical stability in the end. What's also quite interesting is if you look at this picture here, you will see that there are spatial differences in the bone mass. You have much less here on the superior level and more on the inferior level. This is also related to the biomechanical situation in the vertebra. And it's not necessarily age-related. You see that already also in young people. But what you also see is that these areas that have less bone structure, these are the areas that are mostly affected by vertebral fractures. Now, another structure that is very important for the biomechanical situation are the ligaments. They are very important, obviously, in stabilizing the spine, but also in motion guidance. But then they also have one additional role that not so many people know about or are aware of it, and that is proprioception. Basically what that means is that the ligaments have little nerves growing in, and these nerves are very important in being able to sense how many loads actually occur, and then tell the muscular neural system how to respond to these. So that is a very important aspect for function, even so it's not macroscopically very obvious. And all of the ligaments in the spine, as you know, there are a lot of different ones, they all play a role, a contributing role to the stability and the mobility. And there is a very nice study that I just briefly wanted to show you down here from the group in Ulm, from Heuer et al. It's an in vitro study, and what they did is they had human motion segments, and then they started cutting off ligaments one by one in a sequential way. And then they always tested for the range of motion under different loading conditions. So these would be the different loading conditions. And here you start with everything intact, and then you cut away, cut away, cut away, until in the end you do a nucleotomy. And this way you can see that independent of the loading regime, you have a different range of motion, and all of these ligaments contribute in some way to the stability and the mobility. Now, the next structure that I would briefly like to talk about is the intervertebral disc. It's a very interesting structure because it's quite complicated in its tissue composition. You have two different zones that you can see here in this little schematic. You have this inner nucleus pulposus that is very hydrated. Lots of proteoglycans, lots of water, and that basically has, in a healthy person, a gel-like structure. But then you have a highly structured annulus fibrosus. In this picture you cannot really see it, but you can see it nicely here. It's highly organized. Collagen type 1 is the predominant collagen that has this highly organized and hierarchical structure. And you have an orientation that is always alternating by certain degrees, like this and this and this, as you can see here in this picture. And basically, if you would need to imagine it in a real life situation, the intervertebral disc would resemble a little bit a donut or Berliner, like shown here, because it has this inner gel-like part and it has a more structured outer part. And what you also know from daily life is if you have a little hole where the term is actually injected, and then if you press on it, then the term will actually come out, which is basically the resembling image of a disc herniation. Here on this picture, you can see these two structures very, very nicely, the gel-like nucleus pulposus and the organized annulus fibrosus in a very young patient, or not patient, just a person. And what is also important is when you put axial load on the spine and the motion segment, because the annulus fibrosus in a healthy person is very nicely structured and intact, this will actually lead to hydrostatic pressure in the nucleus pulposus because the annulus fibrosus contains the nucleus pulposus. And these hydrostatic pressures, as I mentioned before, can be very high, up to 3 megapascal, depending on what loading you do. Now, one thing that I would briefly like to mention is that these loads, they do not only affect the tissue level and maybe cause a damage in the tissue level if the load is too high, they also have effects on a much smaller scale, and this would be the cell scale. So every loading that occurs, your cells in the respective tissue will sense this mechanical loading and will respond to it in some way or the other. This is called mechanobiology. So it's basically the combination of mechanics and then biology as the readout. And there have been a lot of studies going on more recently because it has been recognized that this is a very important contributing factor to disease development, in addition with, for example, genetics or nutrition, which I'm going to talk about later. And there are different ways of how you can test the effects of mechanical loads on cells. You can do that in a pure in vitro study where you have cells, isolated cells from a biopsy that you might take out in a surgery. And then you apply compression or tension to these cells. But it can also be a bit more complex. And then you would go into an animal model. And there are a few very well-established animal models. Mostly these are rodent models because you have the tail that is very accessible with the intervertebral discs, and you can use that nicely to apply mechanical loads. So this would be an example of a rat compression model, rat tail compression model, where basically you have a rat tail and then you put pins through the vertebra with one intervertebral disc in the middle. Then you have these rings for stabilization, and then you put a loading device on, and then these two rings basically get compressed, and you can do that for basically as long as you want, and you can also change the frequency and the magnitude of the loading, and you can then determine what happens in the intervertebral disc on a cellular and molecular level. The other thing that is important when we talk about the disc as an organ level is intervertebral disc nutrition, in the context with the disc being the largest avascular tissue in the body. Another avascular tissue in the body would be the cartilage in the knee, for example, but the intervertebral discs are actually higher. And because it's avascular, it's also a neural, what that means is that all the nutrients cannot go in via blood vessels as you would have it in a normal tissue, like for example the muscle. They need to go in via diffusion. So the blood vessels here, they end in the vertebra, and then you still have the end plate, and they need to diffuse through the end plate into the intervertebral disc. And of course that means that you have quite a large distance, and you already have cells sitting here in the outside. So all the nutrients that go in, it's like on a buffet a little bit. They are all grabbed, and then the people that are further away, or the cells that are further away, they don't get any food anymore. So these in the middle here, they have very low nutrients, but the nutrients are very, very important, glucose for example, for the cells to maintain their activity. And this is why you have this little picture here. When you go from the outside towards the inside of the intervertebral disc, you actually have lower nutrients, and you also have lower oxygen, because the oxygen goes in exactly the same way via diffusion through the vertebra. But on the other hand, of course, the cells also produce waste products, and they need to go out the exactly same way. So they tend to accumulate in the middle and cannot really get out so much. And one of the typical waste products of disc cells or cells in general is lactic acid. So what happens is that you have an increase, a decrease in the pH towards the middle. So it's more acidic, and this is shown here. And in fact, when you have a degenerated disc, the pH can go as low as around pH 6, whereas in a normal tissue you would normally have 7.4. In the disc it's usually around 7, even in a healthy condition. And this of course also has very high impact when you think about cell-based disc regeneration. For example, with stem cells, you will have to put cells that are not used to such an environment into such an environment. And the question is whether they can actually handle it or not. And this is one of the underlying reasons why it has actually been proposed that stem cell therapy probably will only work in more mildly degenerated discs, but not in the severely degenerated discs where the pH is even lower and the nutrition and the oxygen levels are even lower in the end. And the conditions, the microenvironment as we call it, is very bad for the stem cells. Now if we go down one more level, then we would basically end up in the tissue level. So we're basically now looking macroscopically or microscopically on the tissue. And you can see here on this little schematic how complex the matrix is actually built up. On the one hand, of course, you have the cells. This is here demonstrated. And these cells, they produce the extracellular matrix. For the intervertebral disc, this is predominantly proteoglycans and collagen. In other tissues, you would have other types of matrix. Also, one has to say that collagen and proteoglycan are basically present in almost all tissues. And here, this is a little image of collagen. I already mentioned that. Collagen can be very, very highly structured and orientated. And this little picture actually shows you how nicely that is done. Now, this little graph shows you how these two different matrix proteins, collagen and proteoglycans, are actually distributed in the disc, the nucleus pulposus, the annulus fibrosus, and the end plate. And you will see that you have these components everywhere. But what is different is the ratio of the different components. So in the nucleus pulposus, which I previously said is very highly hydrated, you have more proteoglycans and therefore also more water, because the proteoglycans have negative charges and they attract water. And that allows the nucleus pulposus to have this hydrostatic nature that it has, and also the shock absorbing function. And then in the annulus fibrosus, you have less proteoglycans and more collagen. And what also is important is that you have a different type of collagen. It's actually collagen type 1, this highly organized one, whereas in the nucleus pulposus, you have more collagen type 2, which is less organized and intermingles with the proteoglycans. So very different structure, despite the fact that the matrix proteins are the same. And again, this is a picture that you've already seen more or less, with the two different zones that, again, despite these very similar composition, just a different ratio, looks very, very different. And this is a schematic that shows the same. Here the proteoglycans, it's these brush-like structures with the high negative charges. And here the collagen, here very highly organized for the annulus fibrosis. And the lower picture is actually a histology picture. And what you will see here is an important point. The cells are different, and that makes sense because you cannot have the same cell producing two completely different types of matrix. You have in the nucleus pulposus cells that are more like chondrocytes. They are round, and they produce, like chondrocytes, as in cartilage, a lot of proteoglycans. On the other hand, in the annulus fibrosus, you have fibroblast-like cells. They're elongated, they organize themselves along the fibers, the collagen fibers, and they also produce a fibroblast-like matrix. However, both of these cell types are not exactly like chondrocytes and fibroblasts. They're just fibroblast-like and chondrocyte-like. There are small differences to these. Now, what I mentioned before, and that also comes in here when we talk about the tissue level, especially when we talk about the nucleus pulposus being so hydrated, this hydration is actually very, very important for the nutrition of the disc. We said before that the disc is avascular, and all the nutrients and the oxygen need to go in via diffusion, and also the waste needs to go out via diffusion. And what happens is that because of this high water content, when you move during the day, you actually press out liquid, which you can see here. And then during the night, the nucleus pulposus specifically, because of the high proteoglycan content, takes it up again. You can actually see that in a change in disc height. So overnight, when the water is re-embedded, the disc height will increase by up to 10%, and then during the day, it will decrease, which is also the reason why in the morning you're a little bit taller than in the evening. And with this mechanism, with the loading, this actually allows the diffusion of the nutrients in and the waste products out. It's the only real way how that can happen. So it's very, very important for the health of the intervertebral disc. So loading is not per se bad. It is actually at a physiological level a very good thing. Now, if we go one level lower, then we end up with the cells. And we've already said there are different cell types, nucleus pulposus cells, annulus fibrosus cells, which are chondrocyte-like and fibroblast-like. And this is an SEM picture of a cell in its natural matrix. It is actually a chondrocyte, because I couldn't find an SEM picture of a nucleus pulposus cell that is so nice, but it would look very similar with the highly structured extracellular matrix around, and then the cell sitting in a little bit of a lacuna. And here you don't really see that, but what is important to know, and you probably know that from previous lectures also, the cell is not just a little round blob. It is actually very, very highly structured. You have different organelles in it, and you also have the nucleus in it, which is very important because it holds the genetic material, the DNA. Now, if we talk about cells, there is a word that maybe you're familiar with or not, and it's the name phenotype. A phenotype of a cell means how the cell actually represents itself. It's basically the sum of the cellular processes that lead to a specific type of morphology and a function of the cell, and therefore also what it produces. And in the intervertebral disc, you have very different phenotypes. On the one hand, you have the nucleus cells and the annulus cells, but earlier in the development or at early stages in childhood, you have two additional types of cells that I would briefly like to mention. The one are notochordal cells. These are cells that come from the development from the notochord, and they are very specific because they are large vacuolated cells, and they have very specific biological activity. They are very active in producing matrix, and that makes sense because in the development you still need to produce the matrix for the intervertebral discs. And they're also very, very sensitive to different microenvironmental changes, so very sensitive to load or to low nutrition. And that is probably the reason why, in humans at least, they disappear very early on. So in a human, very young age, just a few months or years, you don't really have notochordal cells anymore. However, you have these cells remaining in a few species, for example, in rodents. And that is why rodent models are often criticized for spinal research, because you do have a different cell type in the end, and the translation to the human situation is actually a little bit questionable. It's not really clear how these cells respond and how much different they respond to nucleus pulposus and annulus fibrosus cells. Then some more recent data that maybe you've heard about is that disc cells also contain progenitor-like cells, so basically cells with a stem cell character. It used to be that stem cells, when you would ask people where they are found, they would always say bone marrow, because that is the very traditional source of stem cells. Then afterwards, fat-derived stem cells became very, very in, because you can obviously get the fat very easily for a stem cell-based therapy. But then it was recognized that stem cells are present in all tissues and also in the intervertebral disc, despite the fact that the disc has such a low healing potential. And the reason is that you don't really have a lot of stem cells anymore in older age. And these cells are very specific because they have a very typical marker expression profile. This is actually a study that was done in Japan. It's a Nature study, so very rare that in our field there are Nature publications, and they described these progenitor-like cells as Tie2 and GD2 positive, so these are the surface markers. And the idea is that maybe one can actually use these stem cells for regeneration, because they are on a certain level already used to the disc microenvironment, but they still have this great capacity of progenitor-like stem cells, progenitor-like cells or stem cells with high matrix formation and so on. Now, when we talk about cells, obviously we also have to talk about cellular activity. And cellular activity means what the cell does. It's on the one hand the synthesis of matrix proteins, extracellular matrix, but it's also waste excretion and generation of energy, division, all of these aspects. And in the intervertebral disc, the most important parts and that you have to understand is that cells produce, on the one hand, matrix proteins. They also produce growth factors which stimulate matrix protein synthesis, but also inhibitors of enzymes. But on the other hand, they produce enzymes that will actually degrade these matrix proteins. And a lot of times people think that the enzymes are the evil ones and the matrix proteins are the good ones, but you actually need the combination of both because you want to remodel your matrix constantly so that it stays young and healthy. But you need a certain balance, and this is what is shown here. If you have a balance between the anabolic and the catabolic processes, you will actually have a healthy disc. But then if this balance tips towards catabolism, you will end up with a more degenerated intervertebral disc. And the same is true for all different types of tissue. So this is a concept that is true throughout the body. Now, obviously you do know that age-related changes of the spine are very, very crucial. We've already talked about back pain and how much that costs and what a high prevalence it has, and the fact that it mostly affects the working population. So understanding these pathological processes is really crucial to be able to develop novel treatment options. And I just want to briefly show you this little picture of a very healthy spine with nice intervertebral discs, also still nice vertebra, and then here on the other side the extreme case where it's severely degenerated, basically with a complete loss in disc height. And you can of course imagine that everything in between also happens, and you can imagine that this can play an important role in disease development and pain development. You have age-related changes, of course, also in the vertebra. We already discussed that briefly with the loss in bone structure, osteoporosis, for example, happening, which again is a little picture here, left side healthy, right side diseased. And then here on the intervertebral disc, in a bit more detail, the different grades. There are different grading methods that you can have. This would be Thompson grading with the healthy one here. And then you have up to the complete disc height loss, fissures and clefts in the annulus fibrosus, all different stages. You can also grade intervertebral disc degeneration not only macroscopically, you can also go microscopically. So this would be histology. It's a grading score that is called histological degeneration score, which was actually created by Norbert Bose a few years back, where you look at specific things that would happen on a microscopic level during degeneration, such as cell cluster formation. So this would be the normal. This would be cell clusters. Probably what the tissue does or the cell does, they want to counteract the degeneration process and try to make more cells. The only problem is that these are not really functional, but you do see these unfunctional clusters then. You also have mucosaccharides that deposit, which are stainable, so you will see these quite easily, and also cell death happening with more degeneration. And all of that you can put together and then create a score of how severely degenerated the intervertebral disc tissue is. Now, this is something that we already talked about. I just wanted to bring it up again because we talk about disc degeneration, and the question is, what is the cause? What we do know nowadays is that genetics has a huge impact. That was not known for a long time, but then over the years it became more and more evident that genetics is probably the most pronounced contributor, and then you have additional contributing factors such as the bad nutrition or mechanical load if it's hyperphysiological, or static, for example. So everything that is outside what we call a safe window of loading. Now this is one of the last slides that I want to show you because it's very important to understand pain development in the disc. Of course, if you have a disc herniation and the material, the extruded material, compresses directly the nerves, spinal nerves, and irritates them, that is a very clear pathway of pain development. But you also have patients that have disc degeneration and they have pain, but they don't have a herniation. But you also have the same ones that show exactly the same MRI picture, but they don't have any pain. And the question is always, why is that? And probably what it comes down, this is kind of the latest research now from the past years, is that it comes down to the cytokine levels that each of these patients produces. In the painful patients, you have more cytokines. I will show you a table of the most relevant ones on the next slide. But what is important to understand is that we've talked about this, the disc is avascular and it's aneural. Only at the very end you have a few nerves growing in. But then when the degeneration happens, you have a loss in proteoglycans, and that allows the nerves to grow in. And you also have clefts and tears happening that also allows the nerves to grow in. So they go in more towards the middle or more to the inside. And then when the cells produce cytokines and they are, with the daily physiological loading, excreted out of the disc, they will actually hit the nerves and can irritate them directly. So this is the concept, the current concept, how degenerative disc disease and discogenic back pain in a case of only degeneration but not herniation actually occurs. This is a little list of, on the one hand, the different matrix degrading enzymes that are upregulated during degeneration and will lead to this matrix degradation that allows the nerve infiltration. And on the other hand, here a list of the very different pro-inflammatory cytokines that are known to be present in the intervertebral disc. And some of these are currently being treated as candidates for new therapeutic approaches, where you would try to either directly inhibit the synthesis of these cytokines or you would try to neutralize them in some way. So basically a non-invasive treatment where you would inject something into the disc as a slow release system, hoping that you can reduce the inflammatory cascade and therefore reduce pain development without needing a surgery, or at least being able to postpone the surgical approach. And this is the very last slide that I want to briefly show you on a very new concept that is very in, and you might see that in a few publications or hear people talking about. So I wanted to bring in this word inflammaging. It is basically the combination of aging processes with inflammation. This little figure shows you exactly what we've been talking about. You have the degeneration happening, you have nerves growing in, you have cytokines. You also have interaction between the local level and the systemic level. You have the different aspects that lead to degeneration, but also to more cytokine production. And the intervertebral disc is actually the prime example for inflammaging, because the degeneration is an age-related process, but in certain patients it is combined with the inflammation and then leading to pain development. So as I said, this was the last slide that I wanted to show you. Here are some of the core messages that I'm not going to summarize again, but it might help you when you want to just get the brief concepts again to study for the exam. And with this, I would like to thank you for your attention.